In modern biology, the color of blood is not considered a mystery. Hemoglobin, an iron-based protein, binds oxygen and produces the characteristic red color associated with human circulation. This model is treated as universal among vertebrates. It is efficient, internally consistent, and taught as settled knowledge. Within that framework, any deviation is usually classified as a pathology, a linguistic metaphor, or a misunderstanding. History does not fully conform to that clarity. Across ancient, medieval, and early modern records, there are repeated references to individuals described as having blue blood, in a literal, physiological sense. These are not social metaphors for nobility. They are not symbolic descriptions of status. The texts describe observable traits: blood appearing blue or bluish-green when exposed, wounds lacking the expected deep red coloration, and these characteristics being treated as inherent rather than diseased. Under the current biological model, this should not happen. Hemocyanin, a copper-based oxygen transport molecule, turns blue when oxygenated. It exists in mollusks, crustaceans, and certain arthropods. It does not exist in humans, according to every accepted framework of human physiology. Human genetics, immune response, and circulatory architecture do not support a functional hemocyanin-based system. That leaves any historical account of blue blood in humans facing an immediate dismissal, metaphor, error, or fabrication. The problem is that these accounts do not appear in isolation. They emerge independently, across different regions and eras, often without direct cultural transmission. Late Greco-Roman medical texts, medieval European monastic records, and early colonial medical observations describe similar anomalies using different languages and conceptual frameworks. The contexts differ. The motivations differ. Yet the physiological details remain strikingly aligned. That is not what we normally expect from a purely mythical construct. Some sources describe blood as unusually pale. Others explicitly note a blue tint. Several emphasize that the coloration becomes more apparent when the blood is exposed to air or cold conditions. These details add no symbolic value. They do not strengthen allegory. They resemble attempts to record observation rather than to transmit belief. Within modern medicine, the most common explanation points to rare blood disorders such as methemoglobinemia, conditions that can give skin and blood a bluish appearance. But these disorders are typically associated with severe symptoms, reduced oxygen delivery, and shortened lifespan. They do not align with descriptions of lineages persisting across generations, often portrayed as physically resilient or unusually tolerant of environmental stress. This is where the asymmetry appears. If this were a rare pathology, why do accounts suggest hereditary stability? If it were observational error, why do the descriptions converge on the same physiological triggers? If it were social metaphor, why is it tied to specific interactions between blood, air, and temperature? Modern science tends to close questions quickly when observations conflict with established models. In most cases, this is justified. But it also creates blind zones, areas where anomalies are too weak to overturn a framework, yet too consistent to vanish entirely. The historical notion of blue blood occupies that zone. No biological samples remain. No genetic material exists to confirm or deny the presence of alternative oxygen-binding mechanisms. What survives are descriptions, distributions, and repetition. They are insufficient to establish a claim, but they are also insufficient to be dismissed as noise. This places a controlled pressure on a foundational assumption: that human physiology is fully mapped at its most basic level, that all meaningful variation has been categorized, and that no substantial gaps remain. Science rarely operates that cleanly. Its strongest models are built on surviving data, not on what has been erased by time, disease, conflict, and selective record-keeping. In this case, if relevant evidence ever existed, it did not endure. The question, then, is not whether humans once possessed hemocyanin-based blood. The question is what value should be assigned to a persistent anomaly that does not fit the current model, yet refuses to disappear from the historical record, and whether our certainty comes from complete understanding or from the absence of enough data to sustain further doubt. When individual anomalies repeat, attention shifts from the event itself to its distribution, not whether something happened once, but where it appeared, how often, and under what conditions. In the case of historically reported blue blood, the pattern is neither random nor evenly dispersed. That alone matters. The accounts cluster. They are disproportionately concentrated in specific geographic corridors, highland regions of Central and Southern Europe, isolated mountain valleys, coastal enclaves with limited gene flow, and communities described as endogamous over long periods. These are not population centers. They are margins, places where genetic drift operates differently, and where unusual traits, if neutral or mildly advantageous, can persist without being rapidly diluted. That distribution does not prove anything, but it does not behave like folklore. Purely symbolic myths tend to spread along trade routes, religious networks, and centers of power. They propagate horizontally, not vertically, through bloodlines. The blue blood descriptions do the opposite. They remain localized. They recur within the same families or closed communities, and they fade when those communities dissolve. That is not the expected behavior of metaphor. Another pattern emerges in how these individuals are described physically. Across sources, they are often associated with pale skin, intolerance to heat, and a preference, or physiological need, for cooler environments. In several medical accounts from early modern Europe, physicians remark on patients whose blood color appeared abnormal, but whose symptoms worsened in warm conditions and stabilized in cold ones. Under standard hemoglobin-based physiology, temperature affects circulation, but it does not alter blood color in this way. Under hemocyanin-based systems, temperature sensitivity is a known constraint. Oxygen binding efficiency changes more dramatically with heat. This parallel does not establish equivalence, but it is an alignment that should not exist if the accounts were entirely fabricated. That alignment repeats elsewhere. Reports describing these individuals often note slow clotting, unusual wound appearance, or blood that darkens only after prolonged exposure. These traits appear sporadically, not universally, but often enough to suggest observers were tracking something specific rather than inventing freely. Importantly, the observers disagree on interpretation, but not on description. Disagreement paired with descriptive consistency is a non-trivial signal. If this were a single misunderstood condition, modern medicine would likely have absorbed it into a known category. If it were multiple unrelated conditions, the descriptions would diverge more sharply. Instead, the pattern sits uncomfortably between those explanations. Similar, but not identical. Recurrent, but not universal. The temporal distribution deepens the problem. Mentions increase during periods of direct observation, times when physicians, clerics, or officials encountered bodies directly through warfare, plague, or rudimentary medical practice. They decline sharply after the standardization of modern medicine, centralized record-keeping, and the reduction of isolated populations through urbanization. That decline is often interpreted as progress, better knowledge replacing superstition. But it is equally consistent with another process, the disappearance of the populations themselves through assimilation, mortality, or loss of distinct lineage identity. Patterns can vanish not because they were false, but because the conditions that made them visible no longer exist. There is also a social asymmetry worth noting. Despite later associations between blue blood and aristocracy, early physiological descriptions do not correlate strongly with power or wealth. In many cases, the individuals described are marginal, rural families, minor local lineages, or socially isolated groups. The noble metaphor appears later, after the biological ambiguity has already entered language. This inversion matters. It suggests that the metaphor may have followed the anomaly, not the other way around. From a statistical perspective, the available data are unusable. Sample sizes are unknown. Observer bias is unavoidable. Selection effects dominate the record. No modern inference can be made with confidence. And yet, pattern recognition does not require certainty. It requires internal tension between expectation and observation. Here, that tension persists. If the phenomenon were random error, the errors would scatter uniformly. If it were myth, the narrative would standardize. If it were disease, the clinical outcomes would converge. None of those fully occur. Instead, what remains is a distribution shaped by geography, isolation, temperature sensitivity, and lineage continuity. Each factor alone explains little. Together, they form a structure that resists simple dismissal. Science is comfortable with noise. It is less comfortable with structured noise. And at this stage, the historical record surrounding blue blood behaves less like chaos and more like a degraded signal, one that can no longer be reconstructed, but also cannot be fully erased. When a pattern refuses to disappear, the next step is not to explain it away, but to test the strength of the models used to contain it. In the case of historically reported blue blood, modern science does not lack explanations. It has several. The issue is not absence, it is sufficiency. The dominant framework begins with hemoglobin. Human oxygen transport is modeled around iron-binding proteins, red blood cells, and tightly regulated pH and temperature ranges. Within this system, color variation is possible, but only within narrow limits. Darkening, oxygen deprivation, or chemical alteration can shift tone toward purple or brown. True blue coloration is not predicted. To account for reports that fall outside this range, medicine turns to edge cases. Methemoglobinemia is one. Sulfhemoglobinemia is another. Both can alter blood appearance and skin tone. Both are rare. Both are well documented, and both carry predictable physiological costs: reduced oxygen delivery, cyanosis, fatigue, developmental impairment. In severe cases, shortened lifespan. These explanations work, up to a point. They explain isolated cases observed in clinical settings. They explain accidental poisoning, genetic mutations, or environmental exposure. What they do not explain well is persistence without collapse. They do not explain multi-generational continuity described without escalating pathology. And they do not explain why some historical accounts emphasize functional normality rather than illness. At this point, the model does not break, but it stretches. Another explanatory layer invokes perception. Poor lighting, limited anatomical knowledge, blood observed under unusual conditions, cold temperatures altering appearance. This is plausible. Pre-modern observation was inconsistent. But perception-based explanations tend to produce noise, not structure. They generate variability. They do not easily produce recurring descriptions anchored to similar triggers. Yet the triggers repeat. Air, exposure, temperature, slow color change. These are not random perceptual artifacts. They are conditional observations. And conditional observations imply that observers were noticing differences in behavior, not merely color. Genetics offers another avenue. Rare mutations affecting hemoglobin structure, oxygen affinity, or red blood cell morphology could, in theory, alter blood appearance subtly. Modern genomics has cataloged hundreds of such variants, but none produce a stable blue coloration under normal physiological conditions. More importantly, none map cleanly onto the geographic and social distributions seen in the historical record. This is where explanation begins to thin. Science is optimized to explain what can be sampled, measured, and reproduced. In this case, there is nothing left to sample. No preserved tissue, no verified remains, no controlled observations. The phenomenon exists only as residue in texts. That places it outside the domain where modern models are strongest. This does not make the phenomenon real, but it limits how confidently it can be declared unreal. Another constraint lies in disciplinary boundaries. Hemocyanin is treated as categorically non-human. It belongs to invertebrate biology. As a result, the possibility of partial expression, non-functional remnants, or evolutionary dead ends within human physiology is rarely explored. Not because it is impossible, but because there is no surviving evidence demanding that exploration. Absence of demand becomes absence of inquiry. Evolutionary biology adds further pressure. The prevailing model assumes a clean divergence: hemoglobin-based systems in vertebrates, hemocyanin-based systems elsewhere. This division is elegant. It simplifies evolutionary narratives. But evolutionary history is not obligated to preserve elegance. It is filled with redundancies, failures, and transitional forms that leave no descendants. The problem is that transitions without descendants leave no data. If a transient physiological variant emerged in a limited population and later vanished through selection or assimilation, modern science would have no mechanism to detect it retrospectively. The model would remain intact, not because it explained the anomaly, but because the anomaly left no testable trace. This distinction matters. Scientific explanations often conflate "cannot be tested" with "did not exist." Methodologically, this is unavoidable. Epistemologically, it is incomplete. The boundary between those two statements is where historical anomalies accumulate. There is also a linguistic constraint. The term blood itself collapses multiple components into a single category: plasma, cells, proteins, pigments. Pre-modern observers did not differentiate these layers. They described what they saw. Modern science differentiates them precisely, but only when samples exist. Without samples, modern terminology cannot be retroactively applied with confidence. So the explanations remain probabilistic, not definitive. Each model accounts for part of the record. None accounts for its full shape. This does not indict the models. It defines their limits. Models are tools, not mirrors of reality. They compress complexity. They discard edge cases. They perform best where data density is high. Here, data density approaches zero, and yet the anomaly remains present enough to require management. It is explained repeatedly, confidently, and briefly, often in footnotes or dismissive clauses, not because the explanation is strong, but because the cost of leaving it unresolved feels higher than the cost of oversimplifying it. That response is human. It is also diagnostic. It reveals where confidence is doing work that evidence no longer can, where explanation is used not to illuminate, but to stabilize belief. This does not mean the belief is false. It means it is under stress. At this stage, the responsible position is not to propose alternatives, but to acknowledge boundaries. To say that current models explain many things exceptionally well, and that they explain this particular historical pattern only partially, indirectly, and with assumptions that cannot be verified. Science does not fail when it reaches its limits. It fails when it denies that those limits exist. And in the case of historically reported blue blood, the limit is not biological plausibility alone. It is the absence of recoverable evidence combined with the persistence of structured descriptions that refuse to resolve cleanly. That unresolved tension is not a flaw in the narrative. It is the narrative. When an anomaly remains unresolved, it does not stay confined to theory. It shapes behavior, classification, and decision-making in the real world. The historical notion of blue blood did not exist in abstraction. It interacted with medicine, social structure, and early biological reasoning in ways that produced tangible consequences. In pre-modern and early modern societies, physiological difference was rarely neutral. Bodies were categorized quickly, often with limited data, and those categories influenced how individuals were treated. Reports of unusual blood were not merely curiosities. They affected judgments about health, fertility, suitability for labor, and, in some cases, survivability under specific conditions. Medical texts from the early modern period describe practitioners altering treatment strategies when confronted with patients whose blood did not behave as expected. Bloodletting volumes were adjusted. Exposure to heat was avoided. Cold environments were sometimes recommended, not as comfort, but as physiological necessity. These choices were empirical, not theoretical. They were based on observed response, not on understanding. That distinction matters. Even incorrect models can generate effective practices if they are anchored to consistent observation. Conversely, correct models can produce harmful outcomes if applied rigidly where they do not fully fit. In this context, the belief that certain individuals possessed atypical blood characteristics altered medical behavior in ways that suggest repeated reinforcement through experience. Social consequences followed. Communities identified as physiologically distinct were often treated as fragile or unsuitable for certain roles. In some regions, they were excluded from military service in hot climates. In others, they were preferentially assigned to environments perceived as less physiologically demanding. These practices imply that the anomaly was not treated as a curiosity, but as a constraint. At the same time, the anomaly could invert social status. As the biological explanation faded and the terminology persisted, blue blood gradually detached from physiology and attached itself to lineage. The physical uncertainty became symbolic capital. What may have begun as a description of difference transformed into a marker of distinction. This transition did not eliminate the original ambiguity. It concealed it. Symbolization is often a way to stabilize unresolved facts. Once embedded in social hierarchy, the phrase no longer required biological accuracy. It required continuity. That continuity carried the term forward, even as its empirical grounding dissolved. The consequence was not clarification, but persistence without scrutiny. There were also biological consequences that are harder to trace. If certain populations were believed to respond poorly to heat, altitude, or exertion, reproductive patterns may have shifted accordingly. Migration choices. Marriage. Restrictions. Occupational specialization. These pressures, subtle but cumulative, can reshape genetic distributions over time, not because of an inherent difference, but because of a perceived one. Perception can be selective pressure. From a scientific standpoint, this complicates retrospective analysis. The very act of labeling may have altered the populations being labeled. Traits may have been amplified, suppressed, or eliminated not by biology alone, but by social response to assumed biology. This feedback loop blurs cause and effect. By the time modern medicine emerged with standardized diagnostics, Many of the conditions that sustained the anomaly had already disappeared. Isolated communities were absorbed. Medical practices were homogenized. Terminology was cleaned of ambiguity. What remained was a stable model and an unstable historical residue. The cost of that stabilization is rarely discussed. When anomalies are absorbed without resolution, they do not vanish. They reappear as metaphors, as linguistic artifacts, or as fringe curiosities stripped of context. The underlying tension is preserved, but relocated. No longer medical, no longer biological. Cultural. This has implications for how science understands its own boundaries. The belief that all meaningful physiological variation has been catalogued encourages confidence, but it also discourages re-examination of discarded data. Historical anomalies become teaching examples of error rather than prompts for epistemic humility. That framing protects the model, but it narrows the field of acceptable questions. In practical terms, this affects research priorities. Phenomena without surviving samples are deemed unproductive. Questions that cannot be operationalized are sidelined. This is efficient. It is also exclusionary. Entire categories of historical observation are removed from consideration, not because they are false, but because they are inconvenient. The consequence is not ignorance. It is selective attention. In the case of blue blood, the real-world impact was not the establishment of an alternative physiology. It was the quiet reshaping of medical behavior, social structure, and scientific confidence around an unresolved irregularity. The anomaly did not need to be true to matter. It only needed to be persistent enough to influence decisions. That persistence left traces. Not in tissue or DNA, but in treatment protocols, social stratification, and the language that still circulates today, detached from its origin. These are not minor effects. They represent long-term interaction between uncertainty and authority, and they underscore a central point. Unresolved anomalies do not disappear. They migrate, from biology to society, from observation to symbol, from data to assumption. The cost of that migration is not error. It is the loss of awareness that a question was ever open. At a certain point, uncertainty is no longer a function of interpretation. It becomes structural. The historical record surrounding blue blood does not thin gradually. It breaks. What follows is not ambiguity, but absence. The most obvious gap is biological. There are no preserved samples, no verified remains with intact circulatory tissue, no blood residues that can be tested, reclassified, or reinterpreted using modern tools. Whatever physiological reality once existed, if any, did not survive the conditions required for contemporary analysis. This is not unusual. Soft tissue almost never does. What is unusual is how completely the gap aligns with the moment when explanatory power would have increased. By the time microscopy, chemical analysis, and later molecular biology could have meaningfully addressed the anomaly, the populations associated with it were already dispersed, assimilated, or extinct as identifiable groups. The timing is precise and irreversible. Beyond biology lies a documentary gap. Many primary records that mention atypical blood appear in sources that were never standardized or preserved systematically. Local medical notes, monastic chronicles, marginal annotations. These documents were not designed to carry authority forward. They were vulnerable to loss, censorship, and neglect. Large portions did not survive political upheaval, institutional reform, or deliberate purging of non-conforming knowledge. What remains is not representative. It is residual. This matters because absence is not neutral. When data disappear unevenly, What survives acquires disproportionate weight. In this case, later dismissals are often better preserved than earlier observations. Skepticism, once institutionalized, outlasts the evidence it critiques. That asymmetry shapes historical memory. There is also a conceptual gap that cannot be repaired. Even if physical samples had survived, the original context of observation is gone. Pre-modern observers did not measure oxygen saturation, copper concentration, or protein structure. They noted behavior, color, response. Modern science requires different inputs. Without them, translation across frameworks is incomplete by definition. No amount of retrospective rigor can bridge that mismatch. The question then shifts. Not what was true, but what can no longer be reconstructed. How much of the phenomenon depended on conditions that no longer exist? Environmental, genetic, or social? How much was contingent on isolation, climate, or practices that have since vanished? Reconstruction requires continuity. Here, continuity is broken. There is a deeper gap still: evolutionary erasure. If a rare physiological variant emerged and then disappeared without leaving descendants, it would leave no genetic footprint. Evolutionary narratives tend to focus on success, traits that spread, persist, and dominate. Failure modes, dead ends, and transient adaptations vanish silently. They are not recorded. They are selected out. If blue blood represented such a dead end, real but unsustainable, it would be invisible to modern biology. Not because it never existed, but because it did not endure. The model remains correct. The anomaly leaves no trace. This creates a false binary. Either something exists and is detectable, or it never existed at all. History does not operate under that constraint. Many things existed briefly, locally, and then disappeared without consequence to the long arc of evolution. Science has no instruments for those. The deepest gap, then, is epistemic. It lies between what could have been observed and what can now be known. Within that gap, confidence becomes a substitute for evidence. Statements become cleaner as data become poorer. Explanations harden not because they are proven, but because alternatives cannot be tested. This is where certainty is most misleading. When asked how much of this phenomenon can no longer be reconstructed, the honest answer is not a percentage. It is a category. All of the elements that would allow definitive resolution, samples, continuity, controlled observation, are gone. What remains are structured descriptions and the absence of a mechanism to verify them. That absence is permanent. The gap cannot be closed by better technology alone. No instrument can measure what was never preserved. No algorithm can recover data that were never recorded. The limit is not technical. It is historical. This does not invalidate modern science. It contextualizes it. It reminds us that scientific models are built on survivorship, on what remains available to measure. When something falls outside that envelope, it is not automatically false. It is simply beyond reach. The blue blood anomaly sits entirely within that unreachable space, not because it demands belief, but because it resists resolution. It occupies a zone where explanation can no longer advance, only stabilize. And stabilization is not understanding. It is closure without completion. Modern science possesses instruments that earlier generations could not imagine. Genomic sequencing, proteomics, high-resolution imaging, and computational modeling allow human biology to be examined at levels once considered inaccessible. In theory, these tools should be capable of resolving anomalies left behind by history. In practice, they encounter the same boundary from a different direction. Genetics offers the most obvious promise. Whole genome sequencing has revealed vast hidden variation within human populations, rare alleles, suppressed traits, regulatory mutations that alter expression without changing core structure. This has reshaped assumptions about what is normal, but it also reveals a constraint. Sequencing requires material continuity. Without DNA, there is nothing to analyze. Without identifiable descendants, there is no lineage to trace. In the case of blue blood, the genetic trail ends before it begins. Proteomics and biochemistry face a similar limitation. Hemocyanin and hemoglobin are structurally distinct proteins with well-characterized signatures. If even trace remnants existed in preserved tissue, modern methods would detect them. But no such tissue survives. The absence of evidence here is not surprising. It is expected. These tools do not fail. They simply have no object. What they do provide is context. Comparative biology has mapped oxygen transport strategies across thousands of species. It shows that nature does not converge on a single solution. Iron-based systems, copper-based systems, hybrid and transitional mechanisms all exist. Oxygen transport has evolved repeatedly, under different constraints, with different trade-offs. This diversity weakens the assumption that one mechanism is inevitable. It strengthens the assumption that multiple paths were explored. Explored does not mean retained. Computational models add another layer. Simulations can test whether alternative oxygen binding systems could, in principle, function within human-like physiology. Some models suggest extreme inefficiency. Others suggest conditional viability under narrow environmental parameters, low temperature, low metabolic demand, limited exertion. These are not predictions. They are boundary tests. They show that impossibility is not absolute. Ancient DNA analysis offers a partial bridge. In some cases, genetic material recovered from remains has overturned long-held assumptions about migration, disease, and adaptation. But even this powerful method is selective. DNA degrades rapidly. Soft tissue degrades faster. Blood chemistry leaves almost nothing behind. The window where such data could have survived is narrow, and in this case, already closed. Modern tools illuminate what might have been missed. They show how much variation can exist without being obvious, how traits can persist quietly under specific conditions, how quickly they can vanish when conditions change. These insights do not confirm historical accounts. They contextualize them. They also expose a methodological bias. Science advances by accumulating positive evidence. It is less equipped to reason about negative spaces, about patterns that disappear before measurement. When tools fail to detect something, the failure is often interpreted as negation rather than limitation. In this case, the limitation is structural. What modern science can say with confidence is narrow. No verified human population currently exhibits hemocyanin-based blood. No known genetic pathway supports it. No biochemical evidence survives. These statements are strong. They are also bounded by present-day observation. What science cannot say with equal confidence is broader. It cannot rule out transient variants that left no descendants. It cannot reconstruct physiological states that existed briefly, locally, and then vanished. It cannot retroactively test observations made before instrumentation existed. This asymmetry matters. It means that modern tools resolve the present far better than the past. They clarify what is. They struggle with what was, when what was left no measurable residue. The answers they provide are therefore partial by design. Partial answers stabilize models. They do not complete narratives. In confronting historical anomalies, modern science does not deliver revelation. It delivers boundaries. It shows where inquiry must stop, not because curiosity has been satisfied, but because the material conditions for further inquiry no longer exist. That realization is itself informative. It suggests that some questions persist not because they are profound, but because they are untimely. Asked too late. After the evidence has already been erased, the tools arrive. The phenomenon is gone. What remains is a refined understanding of absence and an acknowledgement that even the most advanced instruments do not eliminate uncertainty. They simply relocate it, away from speculation and into the structure of what can no longer be tested. At the end of this inquiry, nothing closes. That is not a failure of investigation. It is its outcome. The historical idea of blue blood remains suspended, not between belief and disbelief, but between evidence and erasure. What can be stated is limited and precise. Human physiology, as it exists today, does not support copper-based oxygen transport. Modern biology has mapped the mechanisms of blood with extraordinary resolution. Within that map, there is no place for hemocyanin functioning in humans. This knowledge is strong. It is not provisional. What is less stable is the inference drawn beyond that boundary. The historical record contains structured descriptions that do not fully dissolve under dismissal. They are not proof. They are not data in the modern sense. But they are not random noise either. They occupy a narrow zone where observation occurred before explanation, and where explanation arrived after the observations could no longer be tested. That sequence matters. Certainty is often produced not by complete understanding, but by temporal advantage. When the tools of analysis arrive after the phenomenon has vanished, the model fills the gap by default. The absence of counter-evidence becomes evidence of absence. Methodologically, this is unavoidable. Epistemologically, it is incomplete. The question, then, is not whether blue blood was real. The question is how often similar anomalies have existed at the margins of knowledge, shaped behavior, language, and belief, and then disappeared without leaving a trace that modern science can interrogate. How many conclusions rest not on exhaustive explanation, but on the quiet convenience of missing data? In this case, the most defensible position is not speculation, it is restraint. No alternative physiology needs to be proposed. No suppressed lineage needs to be imagined. The anomaly does not demand replacement narratives. It only demands acknowledgement of a limit, that some historical observations cannot be fully reconciled with present models, not because the models are wrong, but because the past is incomplete. This tension does not weaken science, it defines it. Science advances by stabilizing uncertainty, not by eliminating it. Models become reliable by working where data are dense and repeatable. Where data are sparse or absent, confidence often persists out of necessity, not proof. That distinction is easy to forget. The legacy of blue blood is not a hidden truth waiting to be revealed. It is a reminder of how knowledge solidifies around what survives. What disappears does not argue back. It leaves only asymmetries between what was seen and what can be tested, between what was described and what can be verified. Those asymmetries do not resolve. They endure. And in that endurance lies the final pressure on belief. The recognition that certainty often grows strongest, not at the center of understanding, but at its edges, where questions can no longer be asked, and where the absence of data becomes indistinguishable from explanation itself.